recording here. So we are live and recording. Real quick, I always like to kind of start off with what's going on in the class. Remember that we got 4 more days for the exam. I had to extend it because of issues with Dashboard. Dashboard is working now. Again, um, I had a couple days where I couldn't do anything. It just would not load. Yesterday, it did finally load. I had it trying to load it for 15 minutes on Monday, and it wouldn't do a thing either. So, but it is working. Unfortunately for us, fortunately for you guys, unfortunately for me, that it did allow students, though I had initially set it to have proctoring. Two students did get to take it without the proctoring, and that wouldn't be fair to make everybody else take it without it, and it wouldn't be fair to make those students retake it, because then they would be, whichever way they get in, they get double advantage. So really, the only fair thing is on this exam is to not proctor it. Lockdown browser will prevent you from using your PowerPoints and stuff. So anyway, just wanted to make those things. What we're going to be continuing on. Last class, we talked about the neutrophils, and I talked about what they indicate. Presence may indicate. The indications are something I like to ask on a lot of these. And not every one of them can we really, because they're so generalized what they do. Lymphocytes, for example. Lymphocytes, when we see a lymphocyte here, now remember, this is I always like to say it has a naked nucleus. And what do I mean that it has a naked nucleus? Is you can just barely see this thin halo of cytoplasm around its very large nucleus. This guy, so he has a large nucleus, and then there's a thin halo of cytoplasm around it. So I always say he has a naked nucleus. You can almost not tell that there's anything around the nucleus. It is an agranular leukocyte. Remember that Ben loves money. Ben granular loves money. LM, those are the agranular. Lymphocytes are agranular. Now you could see some granules, but there's few stained granules. Now there's actually 3 types of lymphocytes that we have in a body. It depends on how they get activated, but we have what's called T cells. Now T cells, they behave very differently. They focus only on cell-mediated immunity. For example, they work between other cells, like they go between macrophages and B cells. They may attack a virally infected cell, or at least once they are stimulated to become cytotoxic, they can. And so they always work between cells. So we call them cell-mediated immunity. They can attack cells, or they could actually go between cells. And I really would like to clean that up a little bit on my notes, because they don't just attack cells. When a T cell gets activated to become a cytotoxic T cell, it does. But like a helper T cell, suppressor T cell, these kinds of cells, they are different. Now, B cells, they will practice what we call humoral immunity. They wage their fight in the body fluids. They will release antibodies into the body fluids. So what they do is, these are cells, when they grow up, they can become a cell called a plasma cell. And the plasma cells are little antibody factories who pump out antibodies into the plasma, and that ultimately make it, or into the body fluids like the extracellular fluids elsewhere, the interstitial fluid, and help fight infection. And then our natural killer cells are called NK cells. Natural killers, they kill abnormal tissues like an abnormal cell, cancer tissue, cancerous tissues, and they help to indicate what we refer to, guys, as that there is an infection present. Because when they go high, this indicates that I've got something that I need to fight. So I may have a viral infection, for example, bacterial infection, generalized infections. These things do show up as well. So you may see elevations in neutrophils and lymphocytes both during the same infections. Now, Monocytes. Monocytes, these are really large. They're actually 2 times bigger than a red blood cell is. They're massive monocytes. Remember, massive monocyte. They are agranular. Now, you could see a few little granules here, but there's not as many as there are, let's say, here. Okay? So these granules, few if any, especially when stained, they have the ability to leave the bloodstream. Do you remember what the name of that was called? That a cell can leave the blood and go out to the peripheral tissues? Sounds like the beginning of diaper. Do you remember what that process is called? Diapedesis. They can do diapedesis. And what they do when they undergo diapedesis, they leave they leave the blood vessel, they go out to the tissues surrounding it. They become macrophagic cells. Now remember, macrophages mean you're a big eater. You're a big eater. Monocytes are massive. So these become your big eaters. Okay? Their cells almost look heart or bean shaped. They become a cell referred to as an antigen presenting cell. Now, this is very important for the next unit. Is that antigen presenting cells are cells that what they do is they phagocytize viruses and bacteria, like a bacterial cell. They phagocytize it, they destroy it, but they grab a chunk of it, a protein from that virus or bacteria. They grab it and they stick it on their cell membrane and they take it to a T cell and they say, look what I've got. Look what I found. And the T cell says, oh, yeah, that's very interesting. And I'm going to recognize this as a thing. And if I ever see this, I'm going to know this is the bacteria that you've seen. And it can help recognize and present the antigen. It's one of the cool things about T cells is I will describe that T cells are kind of like when you propose to a woman. If you don't do it just right, she won't see it. She's like, I don't care. Unless you got to get down on your knee and you got to lift it up and you got to hold that ring out and do everything just right so she'll actually be like, yes, I'll accept this. T cells are very picky. They have to have it presented to them. You guys will hear me say, well, if you like it, then you should have put an MHC on it. Now, this is what's going to make antibodies be able to be produced. And we'll talk about this. And they also indicate infection. Now, of course, all these indicate infections, but some of them indicate very specific infections. And I mentioned that bacterial and fungal infections from neutrophils. And here, eosinophils, another one who's got a very specific infection. Eosinophils. Eosin is a stain. You guys may remember that when you did labs in histology lab in A&P 1, and we did the tissues. You may remember a lot of these tissues were pink and blue, or kind of pink and purple, pink and blue. And the reason why it had this reddish, pinkish color and the bluish color is anything that was acidic turned kind of pink or red because there's a stain called eosin. Eosin, if you're an eosinophil, you love eosin. Eosin is an acidic stain, so it's the red stain that's used. So since it loves it so much, we call it an eosinophil because it loves it. It has an attraction to eosin. So we also call them acidophils. Now, they have huge numbers of granules. I mean, look at all these granules. Very deep red granules with a bilobe nucleus looking like Spider-Man over here. Eosinophils, they look like Spider-Man, okay? Now, since they kind of look like Spider-Man, one of the things they're going to do is they're going to attack anything that has antibodies on it. If you coat antibodies on a bacteria or virus, it can attack those things and be like, okay, I'm going to go against that. And they also help me defend against parasitic infections like tapeworms and flukes and nematode worms, a lot of worm parasites, especially like flukes, the so-called trematodes, cestodes, tapeworms, nematodes, the roundworm parasites like hookworms, pinworms, ascaris worms, things like that. Um, so if you've ever heard of those organisms, some pretty nasty infections that can be really horrible. And also, one of the things about them is they release toxic compounds to do this. Not only do that, but when you have an allergic reaction, when you breathe in something you're allergic to, and you start to get the stuffy, sneezy things, the allergic reactions will activate the eosinophils to try to control the spread of inflammation. Since these try to reduce inflammation spread through the body, I call them easy on you phils. They're easy on you because they try to reduce the spread of inflammation. These are what keeps you from basically breathing in pollen that makes you sneeze and then swelling up all over your whole body. Things like that can happen in anaphylaxis. People who have eosinophilic anemia can end up with more widespread inflammations all over their body. They can swell up all over because they have the inability to prevent that spread. Um, so they're easy on you, Phils. They may say that you have a worm parasite. Things like tapeworms, hookworms, pinworms, flukes, like liver flukes, things like that. Liver flukes terrify me. Um, they can steal proteins from your own cells and tissues and stick it on their own bodies and use it like camouflage to avoid your immune system. Always kind of freaks me out, I'm telling you. Um, now, basophils. Basophils, now these guys, they always wear Ugg boots and they drink pumpkin spice lattes all the time because they're basic. Okay. They stain with the basic stain that gives it the dark color. Its name is actually hematoxylin, is its name. Hematoxylin is a very basic loving stain, very bluish. So it takes up so much of the hematoxylin that you will see this like it is just, you can't even see the nucleus. It's so granular. How granular is it? It's so granular, you can't even see the nucleus or the granules. Sorry, that wasn't a joke at all. And it can make it hide it. But if you can see the nucleus, it can be bi or trilobed if you ever get to see it. Now, these guys, these granules are full of histamines. So these guys are bad on you, Phils. They're the ones that cause the spread of inflammation. What's going to happen is when you breathe in a thing that causes you to have an allergic reaction, these guys will receive antibodies from that, and they will dump out histamines and make you get all like stuffied up. And it can cause, like basophils being too numerous, can cause asthma. It's one of the causes of asthma, type of asthma. This is why you go see an immunologist to treat asthma. And so it's responsible for allergic reactions. So I say they're bad on you, phils. Basophils cause allergic reactions. Eosinophils, easy on you, phils. They reduce the spread of allergic reactions. Okay? Very important things to know about. So I put this together that just gives you one-stop shop to see kind of everything these guys do, and the information from my notes. The differential counts, you will not have to know the specific numbers. Remember, you just need to know never let monkeys eat bananas, and that is order from most to least. I will not ask you what percentage, but I will ask you guys to arrange them in most to least for sure. Absolutely. Maybe even least to most. What if I went crazy? I mean, let's do least to most. Ooh. Never let monkeys eat bananas. That is from most abundant to least abundant, okay? Now, one of the things I want to talk about is how do we make white blood cells? Well, this is a process called leukopoiesis. Leukopoiesis, when we hear the word poiesis, we're meaning this is the formation of something. Leukopoiesis, formation of leukocytes, white blood cell production. Remember, I have what's called a hemocytoblast, and it can go in and become a myeloid stem cell to go and produce red blood cells and most white blood cells, or a lymphoid stem cell that can only go and make lymphocytes. And that is my only two things that we have. A hemocytoblast can become either a myeloid stem cell to become red blood cells, platelets, And then neutrophils, monocytes, eosinophils, and basophils, most of your white blood cells. Now, since we're only talking about WBC production, leukopoiesis, we're not worrying about the red blood cells and platelets here. Lymphoid stem cells can only make your lymphocytes, your B cells, your T cells, and natural killer cells, NK cells. Okay. And as we get into that, you'll learn more about B cells, T cells, and stuff like that in the immune system when we get there. Okay. Now, one other thing I do want to talk about with platelets and thrombocytes is the last kind of cell we got to talk about, last formed element, is these guys, when they become a thrombocyte, they're kind of like a disc-shaped cell. And think about you have a plate. You have a plate. And then you drop your plate, and it breaks into pieces. Now, all these broken pieces of plate used to be plate. Used to be part of a continued round disc. But now they're just fragments. Those broken fragments of plate, that is what our platelets are. Our platelets are not actually truly cells. When I actually was interviewing for my job, and I found out nobody else got that, but they did it to me, and I don't know why. One of the people who was on my hiring committee, they asked me, said, "Mr. Lever, do humans have true thrombocytes?" And I said, "No, they're they're just fragments of a cell. They're not really cells. They're cell fragments." And he's like, "Okay." I mean, they asked me. One of them they asked me all kinds of questions. I don't know why. They asked the normal questions everybody else had to ask, but they just for some reason grilled me harder. I don't know why. I don't know if they were they knew me, and I guess I was the only one who was an adjunct there at the time who was applying. So I guess since they knew me and they felt comfortable with me, they felt like they could ask me. And I think that gave me more points on my application because there was a PhD applying. And I guess they probably why they did it, maybe. I don't know. I'll have to ask one day. Um, so I'm glad for that. Now, what they do is these fragments contain a lot of enzymes, proenzymes, and things like that that are there for blood clotting. But they have proteins, but they do not have a nucleus in them. So the plate that we have, the big plate, the big dish that you broke, is called a megakaryocyte. And the megakaryocyte, we dropped it, bam, and it smashed. And all the pieces are platelets. And we call these pieces platelets once we activate those platelets, okay? So they're called cell fragments. Now, only you have, of all the platelets you make, 2/3 of them are in your blood right now. But there's a third of them that are actually there in your spleen, just waiting. And the spleen holds a reserve of about a third. And it's kind of like if you guys are fighting a war and you start to lose too many soldiers in the war, you call up some reserves to help resupply your soldiers on the front. Well, that's what the spleen does. The spleen holds the reserve units of platelets. About a third of your platelets stay in the spleen. And if I get really badly injured, get a really bad cut, we dump them out. We can release them then, and they can help me do that. Now, how do we make a platelet? Well, there's another hormone released from the kidney, just like how erythropoietin stimulates red blood cells. We actually call it sometimes erythropoiesis stimulating hormone. Thrombopoietin, remember when you hear the word like poietin or poiesis, we mean this is a thing that makes this happen. Thrombopoietin is the hormone that causes platelets to actually form. And these guys can go on to form your blood clot. We actually call it a platelet plug. Now, if you have too few, what we call thrombopenia, remember, if a number is tenia, it's something penia. So if the level of thrombocytes becomes tenia, it's thrombopenia. And that's low numbers can cause excessive bleeding when you don't want to bleed. Thrombocytosis is when the number's the mostest. And that is high numbers and has unwanted blood clots. Neither one of those are good. You want clots when you need clots and only where you need clots. You don't want clots where clots don't belong. Unwanted clots can kill, and they do kill. Now, platelets like to party. Let's talk about platelet parties. No, I'm not talking about Anthony Jeselnik's shark party. I'm talking about platelet party. You guys like, yeah, I get it. You like dark humor, and no wonder, no surprise, you listen to Anthony Jeselnik. But platelets like to party. And when platelets party, what do you do at a party? Okay. When you have a party, you gather together. You get some people together. You call up your best friends, and you're like, let's all get together and have fun. Okay. So the platelets, they gather together. Then they're going to stick together. And then they're going to clump together. It's kind of like, you gather together and you guys get with your best buddies. You get around, you start drinking a little too much. Next thing you know, plump, you all clump together. Now you wake up in the morning in a big pile of people that passed out drunk together at a wild party. So they aggregate. That means they gather together. They aggregate. They stick. They stick to damaged surfaces. We say they have adhesiveness. If a surface of a blood vessel is damaged, they can stick to it. And then they could clump together, okay? So it's like at a party, you get all the people you know together. Some of those people are your best friends, so you like to stick in one place together. Then you guys all kind of clump together, and you just stick together. It's kind of like you go to a friend's party. You go with a friend, and you don't know the person who's putting on the party. You only know the person who you're going with. And then you just get left alone at the party, and you're like, I don't know anybody here. This sucks. You're like, oh gosh, it's like me almost every party I ever went to. That's why I never liked parties very much. I didn't know anybody else. I only knew that one person. Because that was always like the person was like, I had like three really good friends. I'd rather have I always like to have small numbers of very close friends instead of a whole bunch of people that I really don't give a crap about. So here is a table that goes through all the formed elements, their basic things about them, what they do. I always like to put those in for my students. I think it helps a lot to have really good look up tables and things. All right. So I'm going to tell you guys on this slide, when it comes to the formation of blood cells, I used a test on it. Right now, we know this is not entirely right now. We've actually learned some things since our book was written, and we know this is not accurate. And I'm going to tell you guys that what's on this side is not entirely accurate, and I'm not going to be actually testing on majority of what's on here now. So don't worry about memorizing these. I believe I took it out of this chapter review. If I did, mark it out, because by the time you guys get into the field, this is not accurate anymore. It's not been published. We just know the latest research is suggesting that it's far more simple. It's just that we misunderstood some things. New modern techniques are showing that the gene activations of those cells are not, they're just cells who are undergoing a process to change, but they are not themselves a unique cell just yet. So this is actually probably more simple. What I'm going to say is you need to know that all cells come from a hemocytoblast, and that red blood cells, white blood cells, and platelets all derive from a myeloid stem cell. Lymphocytes are the only type of white blood cell that has a lymphoid stem cell in its history. And that's the only one. Now, I used to have them learn these, and I tested on it, and I spent all these really stupid time making stupid mnemonics like Randy, his Majesty probably eats rotten manure. Wilma, her Majesty probably makes many bad latkes. Everybody's their Majesty, but Larry, who has lost lollipops lately. And these are ones, I mean, you can see this is my stupidity, I guess, in my mind where I come up with dumb mnemonics all the time. But everything is suggesting this is absolutely wrong. And we don't know yet fully. It's just that we know it's wrong. And it'll take time for the books to catch up to this. And I'm going to say this is wrong. But right now, we do know that myeloid stem cells give rise to everything but lymphocytes. Lymphocytes have a lymphoid stem cell. Now, to make monocytes and granulocytes of the white blood cells, if you want to make them become a monocyte or make them become the other granular stem cells, You need to know that that you use a monocyte colony stimulating factor called MCSF, and if you want to make it granulocytes, you need a GCSF, granulocyte stimulating factor, colony stimulating factor. These are going to be factors that make white blood cells become certain things. But do not worry about things like myeloblasts, myelocyte, band cells, things like that, reticulocyte, any of those things. All right, because we now know it's not accurate. Okay, now here's a very big thing, a very important thing that if you understand when you leave here, you're going to be prepared for your programs. Now, I'll just tell you a quick story, just a real quick story about it, and hopefully I keep it quick. Is so used to before COVID, we'd have programs come, and some of the programs they would contact me because and they contacted me directly. They weren't I know they weren't doing for every professor, but they did. The programs liked my students because they said they had the knowledge they needed to be successful. And one of the programs came by and they were sitting out in the hallway waiting for me to stop lecturing. And then I realized they were out there and I was teaching hemostasis. And I'm not trying to brag on myself. I'm just trying to say that. So the nursing professor comes in and they said, I'm a nurse and I've been a nurse for this long. And they just said, I've just got to say this. They wanted to say to the students, they said, I wish someone had taught me this and taught me like this when I was in school, because when I got in and we were talking about all these different drugs and bleeding and bleeding disorders, I had no clue what was going on. And they said I just was drowning in it. I don't know how I got through it, just skin in my teeth sometimes. They said, but they said, you will need this information. And I know most everybody's going through nursing school. So let's get in first, define the key word of hemostasis. Now, hemostasis simply is the cessation of bleeding. Now, if I tell somebody cease and desist, you need to cease. That means you stop. You stop doing what you're doing. Cease that at once. Stop doing that. Cessation of bleeding means you're stopping bleeding. Now, I remember I had this on a test once. I said, which of these processes is called the cessation of bleeding? Had a student come up to me and said, Mr. Lefford, what does this word mean? And they're pointing to cessation. So I go into my notes and put stopping after it, just so that hopefully it wouldn't have that problem again. But cessation means to stop. Now, there are three phases to hemostasis. We're going to have what's called the vascular phase. We're going to have the platelet phase, and we're going to have coagulation phase. Now, these phases are all three important. One sets the stage for the next, which sets the stage for the next. So these are crucial interlocking things. Now, before we even get started, to make sure the three processes can occur of hemostasis, there are certain molecules you need. Calcium ions and vitamin K are both needed. Calcium ions. Remember, enzymes need ions and vitamins to function. That is two things that can regulate enzyme functions. Is that enzymes need what we call cofactors. Cofactors can be inorganic cofactors like ions, like calcium cation, or organic cofactors we refer to as a coenzyme or simply a vitamin. Both calcium and vitamin K are needed in almost every process of blood clotting and hemostasis. Okay? Now, there are some proteins needed. One of these proteins is going to be fibrinogen. Now, fibrinogen is made by the liver. And if your liver is damaged, you can't make fibrinogen, so you can't coagulate blood properly. The enzymes, most of those enzymes come from the liver as well, actually. These are clotting enzymes, what you would call like factors 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. Okay. Almost all your blood clotting factors, almost every one of them are a type of enzyme. And almost all of them are the same type. They're all what we call serine proteases, by the way. Do not have to know that. That's way in detail. That's some graduate level crap right there, probably is serine proteases. And then having to know the actual organic mechanism of action of how it works and attacks that's a whole different ballgame. Now, what happens? Let's start it. Why would I need to stop bleeding? Well, what causes me bleeding? I damage a blood vessel. I cut myself open. I get a slice, a cut, a scrape, something, and a blood vessel becomes damaged. Now, when a blood vessel gets damaged, cutting it, scraping it open, damaging a blood vessel, its wall of the blood vessel, one of the things that will happen that is very interesting is that when a blood vessel gets injured, those blood vessels have smooth muscles in their wall, guys. And this will cause and trigger what we refer to as a vascular spasm. The smooth muscle will squeeze down, okay, and tense up. And when it does, it reduces the size of the hole at the site of injury. And this reduces blood loss. If the diameter decreases of the blood vessel, this can slow down blood loss at the area of injury. Okay? Now, when this happens, and this is a very important thing to understand about normal healthy blood vessel functions. Now, all blood vessels, they have what's called a lining. Remember that we learned there's a thing called simple squamous epithelium in A&P1. A&P1, we learned about a tissue called simple squamous. And simple squamous epithelium, we said, lines the blood vessels. So if I'm lining blood vessels, the simple squamous epithelium lining of a blood vessel is called an endothelium. Okay? And the endothelium plays a crucial role in preventing unwanted clots. When an endothelium is injured, they'll release chemicals that says, I'm hurt, I'm hurt, I'm hurt. And if they're hurt, it probably means something damaged that blood vessel, and we need to do something about it. The idea that a normal, happy, healthy blood vessel has an endothelial lining and that it is there, and when it is damaged, it changes. But along with it, normal endothelium should play a role at preventing unwanted clots. Now, what happens? Once this contracts, and this happens, the endothelial cells, they'll begin to do something, they'll contract inwards, and they will expose the basement membrane, and these ragged collagen fibers will get exposed. But not only that, they will release chemical factors necessary for clotting. One of the things they'll release is ADP, adenosine diphosphate. And when they release ADP, this is like a cup of coffee for the platelets. Your platelets are going through your blood, and they're going through your blood asleep. They're just sleepy little platelet, and he's floating around. And then some ATP gets him, and it's like you gave him a cup of coffee. He's like, whoa, I'm awake now. Oh, I'm awake. I need to do something. Somebody woke me up. I need to now go in and help seal a wound. They'll make what's called tissue factor that also activates my platelets. They'll make another thing called prostacyclin. Now, platelets are awake. Now, here's the deal. The bigger the injury is, the more time it's going to take to heal it and the more platelets you need. But if I've got a small injury, last thing I want to do is just platelets going wild. This is not Cancun and spring break, and platelets are not on spring break. We don't want platelets to be like a bunch of Amish kids out on the Rumspringa going out there going crazy. You guys, there's probably nothing crazier than it's like when the preacher's kid gets loose for the first time. They're like, oh, crazy, do all the drugs. Okay, and we don't want this to happen. What we want is we want the platelets to just do what they need to do, no more, no less. So we need prostacyclin, which is a type of prostaglandin, by the way. It's called PGI2. We try to prevent the platelets from overreacting. And the endothelial layer releases a chemical called endothelins. Endothelins reinforce the vascular spasm. They will make the blood vessels vascular spasm a little bit more. They also stimulate growth of endothelial cells so they can start trying to rebuild the dike. Okay. And they'll cause more smooth muscle cells to come in to rebuild the blood vessel. They'll cause fibroblasts to come in and rebuild the fibers around the outside of a blood vessel. So they're going to help to heal the blood vessel back up. Now, what happens? Now, I'm going to tell you the name of this thing. Don't worry about the name of this thing unless you really just want to write it down somewhere. But it's called, the name of the molecule that endothelial cells release that make it sticky is called von Willebrand factor. There's a very important bleeding disorder out there called von Willebrand disease. It's named after a guy named von Willebrand. And von Willebrand factor, VW factor, it goes out and it sticks on your cells. And when a platelet comes by, remember, he's coming by, he's sleepy. ADP and tissue factors are like a good cup of coffee. It wakes him up. Now he's awoke. He will grow these hooks on him, little protein hooks. And he's going to come by and snag on that von Willebrand factor. Because platelets, they stick together. They become adhesive to damaged surfaces. So they gather together, they stick, and they clump to make and seal the wound. Now, would you agree that vascular spasm is already reducing blood loss? Then what we do on the damaged surface, platelets stick to it to further close off the holes. And then the endothelians come in, and they're going to try to regrow the damaged blood vessel. Okay? Now, one of the things that does stimulate platelets that our book doesn't talk about is, oh yeah, I do talk about it a little bit. Endothelial cells, when they actually touch exposed collagen fibers of the basement membrane and they're sticking out, they hit it, they go, ooh, that's not supposed to be there. It's kind of like when you're swimming in the lake and your leg touches a branch and you're like, oh crap, what is that? And it really just kind of freaks you right out, you know? Okay. Now, once the platelets have stuck and the blood vessels are spasming to try to reduce the wound, we go into platelet phase now. So we already know that vascular spasm is reducing blood loss. Damaged endothelial cells are releasing ADP to wake up the platelets, and releasing tissue factors to wake up the platelets. It's releasing what's called a prostacyclin, which is going to reduce the platelet. We don't want platelets everywhere. Because, like, last thing I want, if I hit my hand on the table, when I did that, I probably caused about 100 micro breaks to my blood vessels right now. But I don't want to cause my whole body to turn into one giant blood clot. How do I prevent that from happening is things like prostacyclin and the fact that only I damaged the endothelial cells there. I didn't damage endothelial cells over here. Okay? Now, platelet phase, once the sticky endothelial cells, by that von Willebrand factor that I was talking about, and the mature platelets grow these hooks, and the sticky grabs it, hooks them in, and they're like, I'm there, and I've been activated, platelets will start to release molecules that activate and attract other platelets. It's kind of like when the first people show up to a party and it is off the chain. It is awesome. This is a party that's like no other party. This one is going to be legendary, okay? This is that legendary party, okay? I mean, you got some good-looking girls there. You got the best drink, good music, everything. I mean, it's vibing, man. It's all good, okay? And you're like, dude, get down here. This party is awesome, dude. Get down here, dude, okay? And that's what the platelets are doing. They're like, man, I'm over here at this guy's place, man. It's a rager. Get down here, man. Oh, look, that dude's doing a keg stand. Oh, man. And it's like, this is how awesome it is. And you call people down, man, it's awesome. Get down here. Okay. That's what the platelets do during platelet phase, is they're telling the other platelets that come through, dude, it's awesome. Get down here. We're having a platelet party. Okay. And so you got a gal bringer, if you got a gal bringer, then we all have a humdinger. Okay. So what we do is the platelets, when they're attached there, platelets adhere and they release molecules that activate more platelets. Because what do when the first people come to the party and they see how awesome it is? They call their friends, get down here, man, it's off the hook. They come in and they party with you, and that's the platelet aggregation. And we form what's called a platelet plug. Platelet plug starts to form. So we have now formed what's called a platelet plug. We plug the hole. The wound is sealed. So basically, you guys remember when Honda invented the self-sealing tires, that if you run over a nail, it'll reinflate itself and seal the tire so you never go the run flat tires and stuff like that. Well, guess what? We've had that. This has existed long before that. I don't know how long, but it's existed long before that. Definitely, I mean, before humans. the Now, but then what happens is the platelet phase. So how do we get other platelets to join our party? Well, when platelets arrive to the site of injury, they get activated. It's like, dude, I'm an activated platelet. I'm having a good time at this platelet party. You need to come to the platelet party too. And they come in. Activated platelets release ADP. Now, we already know ADP is like coffee for platelets. So you release it. Now, we release two things. We have what we call SAC granules in a platelet. They release serotonin. And they release, the A is thromboxane A2. So we call them SAC granules, because calcium is also released from it. And that's three of the big things is serotonin, thromboxane A2, and calcium, SAC. The A2, thromboxane A2. And serotonin, they stimulate the vascular spasms to make the muscles squeeze down. They also release platelet-derived growth factor, which also helps the blood vessel heal. It's kind of like the PDGF. It's kind of like when the people come to party and they have a good old time. If they're good friends, they're going to clean up after, so you're not left with a mess, So they're like, dude, we know you trashed your house, but I'm going to help you fix it. Okay, we're going to clean up after it. Calcium is required for platelets to actually aggregate and for coagulation to happen. So this sets the stage for the next bit, okay? The next bit to happen. Now, the next bit, which is actually here, sorry. But we also got to make sure we don't have platelets everywhere. Now, sometimes you want to keep a party contained. The last thing you want it to do is spill out into the neighbors' yards. And it turned, well, depending. That's how Woodstock got started. It just kept growing and growing, and we hardly limited it. But you really don't want your party to start spreading out into your neighbors' yards. You want to keep it there at your place. So you're like, you've got to have some ground rules. Prostacyclin, remember, PGI2, is there to limit the spread. Now, your WBCs, white blood cells, will release some inhibitory compounds to try to prevent the blood clot from spreading through the whole body. And there are enzymes out there who break down ADP to try to prevent ADP from waking up more platelets. Now, one thing I always remember, so growing up, and it's kind of like, this is what these enzymes do. So next door to us, growing up on Possum Trot Ridge, so there was this bit of land that was next door to our log house. And we lived in we had 2 bedrooms and a log house top of a bridge called Possum Trot. And this old guy put a trailer next to us. Real nice old guy. And Mr. Lay was his name. And one night he lets his grandson stay in his trailer. And his grandson throws a party. Well, he's throwing a party, and late at night, we hear somebody says, "What about the neighbors?" And we hear a guy go, "F the neighbors." So about 3 in the morning, after everybody's passed out, hungover, my dad backs his truck up, right up to their window, kicks his truck doors open, puts on Paint It Black, and gets out with his shotgun with double alt buck magnums and starts shooting it around. I mean, you just hear bam, 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 and somebody playing, gonna paint it black. You know, just get out, bam, 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 bam. And they're just stumbling out there drunk and hungover, grabbing their heads out there in their underwear, running out with their clothes in their hands. It was a sight to behold. And that's what these enzymes do. Is they're gonna like, oh heck, you guys think you're gonna keep partying? We're breaking this party up. We're gonna limit your party. So these platelets, they get activated by ADP. They get activated by some molecules called thrombin, and they get activated by collagen fibers. So if we want to prevent that, platelets release clotting factors that lead to the next stage. And that's now what the platelet has done. Is now that I've got the wound sealed, I've got a platelet plug, we now need to coagulate. That is the second most, probably one of the most hated words next to moist, is coagulate. Now, coagulation, it is a process that occurs in plasma. It is when plasma, the fibrinogen in plasma. Remember, if you ever hear the word, if you ever see the letters gen at the end of something, this is a thing that needs to go up in your toolkit, up in your brain, forever brain. I always call it it's got to go up in your forever brain. Is if you see the last three letters gen at the end of anything in class, it means it's not active. It's inactive, and it needs to be activated. We're going to activate it and turn it into fibrin. How do we do that? Through coagulation. Now, there's going to be three pathways to coagulate. To make coagulation is the conversion of fibrinogen to fibrin in plasma. That's all it is. To do it, we're going to take our liquid blood and turn it into semi-gelatinous blood, plasma. There are two pathways that kick it off, the extrinsic and intrinsic pathway. Those kick it off. And then the common pathway, once intrinsic or extrinsic gets kicked off, you can have a common pathway happen. It's kind of like almost anything. There's like it's like I always kind of think about these the intrinsic versus extrinsic pathways. It's kind of like when you're trying to play a stealth mission on a video game and you try to be all sneaky, but you know what's going to end up is you're going to go in guns blazing because I suck at stealth. I am not a good stealth player. I just don't do stealth. I like to go in there guns ablazing. That's just me. Double guns, bam, 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 bam. Okay. Now, extrinsic pathway is an external trauma, like I get punched in the face. Intrinsic is internal trauma, like a cannulus or a catheter gets placed in a blood vessel and it scrapes the endothelium. Then what happens is this kicks off your intrinsic pathway. Now, both of these can kick off the common pathway. Common pathway is initiated by both, only takes one, but both can do it. The extrinsic is faster to do it, but they both will activate factor X or factor 10. And then the intrinsic is kind of there to reinforce the extrinsic. Okay. Now, you only have to know the common pathway. Common pathway is one you got to memorize. I will ask it on the test. It will be on there. But factor X is got to be activated. Now, let's think about it. Factor X has got to be activated. X marks the spot. Factor X. Also called factor 10. Okay? X marks the spot. Okay? Now, I want you to kind of write that because that's what we're going to do. Now, factor X is activated. Now, whether or not you're intrinsic or extrinsic, we're going to activate an enzyme called prothrombinase. So let's do extrinsic. Let's do intrinsic pathways. These two pathways both come in and activate an enzyme called prothrombinase. Prothrombinase gets activated by these two pathways, is prothrombinase. Now, prothrombinase is a proenzyme. It activates prothrombin to become thrombin. So when prothrombinase comes in, and I've got a molecule called prothrombin. Now, you guys will learn in blood tests, there's a thing called prothrombin time. And no, it's not like it's not a TV show. It's prothrombin time. And prothrombin becomes thrombin. Now, what does thrombin do? Thrombin ultimately turns fibrinogen to fibrin. Okay. So what it is factor X is activated by both of these, activates this, and I was supposed to do. Did you see what I did? I was supposed to write this down here, guys. I'm sorry. I was supposed to write extrinsic. I am so sorry. I don't know why I did this way. Mondays have me so tired usually that I just seemed is they activate the factor X, and then factor X comes in to activate prothrombinase. Sorry about that. Now, thrombin is going to be causing fibrinogen to become fibrin. Okay. So we might try to do it like this real quick and see if I can make this a little bit better. So remember, X marks the spot. You have your extrinsic pathway. And you have your intrinsic pathway. And they both activate factor X. Now, factor X can activate prothrombinase. Now, prothrombinase is an enzyme who takes prothrombin and converts it to thrombin. And then thrombin will come in and take fibrinogen to become fibrin. Okay? And this is called your common pathway. Okay? Common pathway. Now, when the fibrin is made, here we have that platelet plug that was made during platelet phase. When fibrin is made, it comes in and it starts to adhere and stick onto the platelet plug, reinforcing the platelet plug, making it more strong. We actually call this one here a secondary platelet plug. In medicine, this is called a secondary platelet plug or secondary hemostatic plug, whereas this is actually called a primary platelet plug or a primary hemostatic plug. Now, does that make sense? That factor X is activated. When we activate factor X by either the extrinsic or intrinsic pathway, factor X activates prothrombinase. Prothrombinase can cause prothrombin to become thrombin. Then thrombin can cause fibrinogen to become fibrin. And the fibrin goes in and goes onto my platelet plug and reinforces it and makes it stronger. Okay? Now, after a while, this is no longer needed. This is no longer needed, and we want to get rid of the platelets and the clot. So the clot needs to retract, and about a half an hour to an hour later, we can start to retract that clot most of the time. And the platelets, they contract, they start to get torn apart, and then what we do is fibrinolysis occurs, where we take an plasminogen. Now, plasminogen is activated in two ways. It's activated by thrombin. Now, we know that thrombin causes fibrin to be produced. And plasmin dissolves fibrin. But plasmin is also activated by thrombin. because plasminogen, which is activated by thrombin to make plasmin, and plasmin actually dissolves the clot. So not only does thrombin make fibrinogen become fibrin, thrombin also activates the enzyme that breaks the fibrin down. Okay? So it causes the clot, the coagulation happening, and then it also causes fibrinolysin or lysis. Now, the fibrin strands get digested. Now, one of the ways to also make plasminogen get activated is there's a chemical I can give called tissue plasminogen activator, or TPA. We call this the clot buster. Now, my brother-in-law one day, he works at a factory, and he hit his leg hard and tried to work for the rest of the day. He got sore, so he went home early. And he's saying it really kind of got where he was feeling like he had a hard time breathing and felt tired. So he was sitting in his chair, and he said he was sitting there, and then all of a sudden, he said it felt like somebody was taking like a knife and running it into his chest. And he was like, I can't breathe, and he was in horrible pain. And well, luckily they called my niece, who is a nurse, and she says, you need to tell somebody that he needs TPA right now. And he was very close to be outside of the window for getting TPA, tissue plasminogen activator. Only 12% of people who got the type of pulmonary embolism that he had survive. Because what an embolus is a blood clot that wanders around. It's a wandering blood clot called an embolus. And it migrates and ends up in the lungs or the heart or the brain, causing pulmonary embolism, causing heart attack, or causing a stroke, or just causing an infarction of any other tissue that it ends up. Wherever the blood vessel goes and it sticks, it causes loss of blood. Now, one of the things is we have proteins in our blood that makes our blood clot very quickly. And that's good if you're living on the savannah and you're running around with spears and trying to eat antelopes. But when we live in a day where emergency medicine, it actually makes it harder for emergency medicine. It was good back in the Neolithic era, very bad now. And I know I've probably made some people mad that I espouse some evolutionary biology ideas as how these things may come about. I'm not saying that you have to accept them. But now, 5 big things to prevent clots. There's anticoagulants the body makes. A good one is called antithrombin 3. Heparin or heparin sulfate. Heparin actually prevents blood clotting by actually taking antithrombin 3 and stimulates it, which inhibits thrombin, which prevents the activation of fibrinogen to fibrin. Thrombomodulin actually activates a thing called protein C. I have a video on this. Protein C actually digests clotting proteins. Prostacyclin, which actually acts to inhibit platelets. And TPA, which we call a clot buster. It actually lyses, it turns plasminogen into plasmin to actually enhance what we call fibrinolysis. So when plasminogen activators come in, it makes plasmin, and plasmin eats up this stuff right here. These are fibrin clots right here. These are fibrin strands. And what we do is we dissolve it, and the blood can flow through a lot better. But this is only needed after the healing has begun and the blood vessel can fully heal. Now, neither of these are on there, but I do want to. Anemia is you don't have enough red blood cells. Inadequate oxygen. There's a variety of reasons why you're anemic. Vitamin deficiencies. There's various kinds of coagulopathies out there, bleeding disorders, where the inability to coagulate. A lot of coagulopathy is caused by liver diseases. As you will see, most every single one of these clotting proteins, most of them come from the liver. So coagulopathies can often be caused by liver diseases or various other things. Hemophilia is a genetically inherited thing. There's actually three types: hemophilia A, hemophilia B, also called Christmas factor because it's named after the patient whose last name was Christmas. I forgot the first name of the kid who got it. It's actually a deficiency in anti-hemolytic factor B or factor 9 deficiency. IX, I always think that IX, I think about Christmas, Xmas, IX, and then what I do is I make that I into a Christmas tree and then put MAS after the X to remember Christmas factor. That's how I remember it, Christmas factor. If you ever have to know it, which you may in patho, is I always think factor IX, which equals 9. And I go IX mus Christmas disease IX9 anti-hemolytic factor B or factor 9 deficiency. Hemophilia C is anti-hemolytic factor C called plasma thromboplastin antecedent. These aren't on the test, but I was explaining a little bit. And now, if you ever have to know for your programs, I gave you a list of all the clotting factors, what they do. So, all right. Now, let's see here. I wish I had. Can you guys tell me what if a patient was having a stroke and I gave them tissue plasminogen activator? Would that help my patient? What is a stroke? Does anybody know what a stroke actually is? Yeah, clot in the brain. When a blood clot goes to the brain and blood supply is lost, you guys, a lot of them may happen in the circle of Willis. Have you guys learned circle of Willis in lab yet? Okay. So I don't know what you guys do in lab yet. See, I'm a little bit behind lab right now because I'm pushing as hard as fast as I can go, and I feel like, but this is a very important thing. We said that tissue plasminogen activator activates plasminogen. What does plasminogen become? Plasmin. And what does plasmin break down? Clots, which is fibrin. So we know that. Tell me what might activate factor X. This is a better drawing than the first one I did. I was like, I should have put it at the top. Yeah, here's what I'll show you what it was. I was actually in my head doing. This is something I'm working on for a video for my YouTube channel. Oh, sorry, I went. Is I'm doing this to help working on most of these. 13 is just not on there, but all the other blood factors from the intrinsic and extrinsic pathway. 7's the extrinsic only has one blood factor, factor 7 to activate 10, so it's quicker because it has less. So, and I always like X marks the spot. Then I go through and I do the it's 12, 11, 9, 8. Why did you pass 10? Because 10's already here. To help students actually remember, and then actually remember some of the diseases and some of the different things. I'm like fit pants, tight collar, loose American shirts, is to help remember the different different things out there. It will make sense after I'm done. I'm putting it together to do coagulation cascade video, but definitely if you this and the other things. But guys, I'm going to tell you, and really, you'll need to know the stuff I said. I'm not kidding. And I was so like that professor said, I wish somebody taught me that. All right, so let's do the thing I know you guys all love to do, and that's take a quiz on this content. And I'll probably try to start a little bit of the heart today if I can. I always get so like when you get me onto coagulation, I'm like, I just start going. I go in. It's like, I don't know, hemostasis has always been a favorite concept of mine. But the things that I taught are all needed in healthcare programs. So you will need to know it. And if I can help you guys understand it. Now, One of those things I would like my students to do if you guys come to study group this week is to draw out the process of um, the common pathway. Is That's one of the things I'd love to have you guys do. Okay. And of course we will have it this Friday. Urge you guys to come. I think you can get a lot out of it. Cassie's been there every week, and so, and you can ask her what she thinks. All right. Oh yeah, and most of the time you're really spending it with the tutor. Let me go in a little bit on the heart today. What I want to try to do is at least play a little catch up. I always feel like I'm behind, which in A&P you always are. Especially if you're trying to teach A&P to prepare people for healthcare programs. And there's a lot of people that said, well, I barely do anything with hemostasis. It's not that important. I was like, what do you mean it's not that important? I was like, if the stuff I'm covering, guys, I'm not kidding you. I've heard too many people say, yes, they absolutely need to know it. Actually, one of the reasons why I put the diseases at the end is a doctor friend of mine. He's actually the guy in charge of my battlefield platoon. But I talked to him a lot about some. Now, I thought a good quote for this one was, a good head and a good heart are always a formidable combination. So you need both. You guys are all smart. Just always make sure you have a good heart in you. If your heart isn't in the right place, if you're going into healthcare for the wrong reasons, I'm going to tell you guys, you will not like it. You're going to hate it. There's nothing worse than somebody who's not in it to actually help patients. You will burn out. You will not like it. That's the only thing that's going to keep you going. Yeah, you have to have a healthy detachment, but you also can't stop caring. The moment you completely stop caring is when you need to quit. But also, there is a healthy detachment needed. Okay. Okay. So remember, I do ask on all these different particular things here, so please pay attention to those. Now, the first thing I want to talk about when it comes to the heart is the heart's a pump. It's a muscular pump. And the heart has to move blood through miles of blood vessels. Honestly, miles of blood vessels. In fact, if I took all the blood vessels in your body and I took them out and I laid them end to end, you'd die. Okay, now your heart's about the size of a clenched fist, okay? As people say, the best way to a man's heart is through the stomach. I disagree with that. The best way to a man's heart is between the third and fourth rib at the midclavicular line. But anyway, now it weighs about 306 grams to 388 grams. Okay. Now a heart beats around 100,000 times a day. Now here's the big thing: is your circuits, and let's talk about heart circuits. Okay, heart circuits. We have circuits, and the heart itself has to be able to do things like the body and also the lungs. Okay. Now, anytime you will have blood and we refer to this as the systemic circuit. And then you will have blood in what we call the pulmonary circuit. Now, we probably know by now there is a right atrium, right ventricle, left atrium, and left ventricle of the heart. And we know that that's how these different circuits are attached. Okay. Now, the big thing that you guys need to know is that your pulmonary circuit, it starts at the right ventricle of your heart, goes to the lungs, and then from the lungs back to the left atrium. And the systemic circuit starts from the body systems. It arrives to it actually comes to the right atrium from the body systems. It leaves the left ventricle of the heart to the body systems and back to the right atrium. Now, the coronary circuit is actually part of the systemic circuit. It's because the heart itself is a body system, part of a body system, part of the cardiovascular system. But it is the circulation that's purely based on providing blood to and from the heart itself, the heart muscle cells, cardiac muscle cells. Okay. So, right here's the thing we really should know. Okay. Is the two different circulations, specifically systemic and pulmonary circuits, what they are, what they supply, how they differ. Okay. Now, I know I only got 10 minutes, but I'm going to use every bit of it I can. Now, the heart. The heart it pumps through miles of blood vessels. It's a hollow muscular pump. It's in the mediastinum, which is the chamber of the thoracic cavity, subdivision of thoracic cavity, where the heart sits. It's between the lungs. It's the area basically between the two lungs, between the two pleural cavities, where the heart sits. Now, in the mediastinum, the heart kind of tilts down to the left, so that 2/3 of the heart is actually left of midline. Now, the apex, which is the so-called pointy end of the heart, it projects downwards to the left, where the base, the broad end, is up to the right. So the heart twists. It is not in the center of the body, so that the right ventricle is the one facing you, because it sits to the left. The right ventricle faces more forward. And this puts it, which is also good because the right ventricle is the one that has the lowest pressures. All right. Now, another thing that happens is the heart has what is called a serous membrane around it, called the pericardium. Now, remember, pericardium surrounds the heart. That is, the membrane surrounds the heart. So if I've got a heart right here, this is my heart. Now, I don't know if you guys know why this is always used for the love heart, but it is representing of the space taken up by two anatomical hearts. That's why it's the symbol of love. You're like, and here we have our body cavity. I thought about using the quote, "The heart is a wild creature, and that's why the ribs are a cage." That was almost the thing I used. So, okay, heart. Serous membrane. So on the heart muscle itself, you have a layer of the serous membrane here. And then we come around, we fold it around, and then we have an outer layer. And the innermost layer here is called the visceral pericardium. And the outermost layer is called the parietal pericardium. That's how I have to remember how to spell parietal as I sing Bad Romance. I'm like parietal parietal. It's like they always say a good romance starts with friendships, but a bad romance begins with ooh rah, ooh la la, something like that. Who knows? I don't care. Now, to make your parietal pericardium, it actually lines the outside of the pericardial sac. It's the outer covering of pericardial sac. It is thicker. It has 3 layers. It has a fibrous outer dense irregular connective tissue layer called the fibrous connective tissue layer, which makes it very hard for it to stretch outwards. This is why we can get congestive heart failure. There's nowhere for the pressure to go outwards. It can only go inwards, and it squeezes on the heart and congests the heart. Mesothelium. Now, we talk about endothelium as the lining of blood vessel. Mesothelium is the simple squamous found inside of your serous membranes, called mesothelium. The areolar connective tissue sandwiched between the two help hold it all together. Now, the visceral pericardium is also called the epicardium because it's upon the cardium, upon the heart. It only has mesothelium and areolar. Remember, mesothelium is the simple squamous epithelium that is associated to all serous membranes of the body. Areolar is loose connective tissues. Now, there is a space between these full of fluid called pericardial fluid. It's between the visceral and parietal pericardium. Now, this fluid normally should have a normal volume. Now, let's say you get pericarditis, an infection caused by one of the things is Streptococcus pyogenes, which is what causes strep throat, Streptococcus pyogenes. And you get that, and it gets in there and infects the pericardium, called infective pericarditis. Pericarditis, one of the things that can happen is the fluid in there builds up, and it can cause problems. Sometimes it also gets inside the heart and grows on the inner lining. Now, the heart has a wall. The heart wall is 3 layers, and the outermost layer of heart wall is called the epicardium, or the visceral pericardium. Then the myocardium is the muscular stuff. When that dies, that's called a myocardial infarction due to or a heart attack. Heart attacks are vascular. Cardiac arrest is electrical. Now, myocardium is cardiac muscle tissues. Then there's the endocardium. An endocardium is the lining of the what's called the endothelial lining of the heart and the areolar tissues that are inside it. It covers all insides of the heart wall, and it covers the valves and things like that, and even covers the tendons, the chordae tendineae of the heart. And it's in there and also can get infective. Strep pyogenes can grow on its surface and become to cause damage to the heart valves, like in rheumatic heart. And also can become, I showed this one time, and I showed this to my students, and I'm going to tell you what happened. One of the students in the class, their mother died of endocarditis, but I had showed this, endocarditis or lasagna. And he was like, my mother died of endocarditis. But I was like, is it endocarditis or is it lasagna? It's like, that's just gross, isn't it? I know I should probably go to heck for that, but okay. So, now the heart. Remember, the base, that's the broad-end. It's at the top. That's where all your great vessels attach, where your aorta and vena cava are all stuck in. The apex is the part that sticks downwards. We refer to that as the inferior part. And the coronary sulcus is the groove found between the atria and the ventricles, which you can find that coronary sulcus. That's where you're going to find like your left and right coronary arteries are sitting in the coronary sulcus. Now, heart muscle tissues, cardiac cells, they have intercalated discs. Remember, intercalated discs are one of the telltale ways to tell the difference between cardiac muscle and other kinds of muscles. Now, what you find here is a very special kind of intracellular junction called a gap junction. Gap junctions are intercellular junctions that are present that let fluid from one cell cross through that hole into another cell. Now, if I let sodium cations from one cell cross into the next cell, I can make the next cell get stimulated. So this allows us to connect our cells chemically, thereby also connecting them electrically. And we attach them using desosomes to hold the cells and tissues together. And this is why the heart can beat in a coordinated way without every cell needing a neuron. And the cardiac muscle cells, remember we said they are branched, single nucleus, and striated. We said they're involuntary striated muscle. So you cannot control your heartbeat. You cannot tell it not to beat, or you can't tell it to beat. It just does itself. Now, you can put yourself in a mental state to slow your heart rate down, but that's the only conscious thing you can do is you can calm yourself down in your heart. Now, as I always like to say, at no time in the history of calm down has anyone ever been calmed down by being told to calm down. That never calms anybody down. Okay, you don't calm down doesn't work. Okay, don't say calm down. Especially guys, never say that to your wives. You know, it's never tell your wives they say calm down. No. All right. I know I won't have enough time, but when we come back, we're going to draw the heart, blood flow through the heart, talk about everything. And then when we get the blood flow to the heart, you're like, oh, it's pretty easy. And we'll draw that out. And next chapter is with what are you guys doing veins this week or next week? When are you doing veins? Next week or this week? This week, okay. Next chapter is circulation and blood vessels, where I talk about the physiology of blood vessels and the basic structure of the walls of blood vessels. But it is that time, so I am going to upload your UJA.